All right, we are assessing the results of our fermentation test. So we used a carbohydrate setup with a durum tube, phenol red broth, and a specific carbohydrate to test. So we can see in this setup that we have some differences. So let's come in a little closer and look. On the left-hand side, we see that the liquid in the durum tube has turned yellow and that there is a bubble present. So this is our E. coli, whereas our Micrococcus luteus, the broth has not changed color and there is no bubble in the durum tube. So our Escherichia coli, as indicated was positive for the ability to ferment this substrate, as indicated by the color of the phenol red broth in the tube. So it has turned yellow, indicating that organic acids were produced and that phenol red changes to yellow under acidic conditions. Also, we can see that little tiny bubble indicating that gas was produced during the fermentation of our carbohydrate, and that was trapped in the durum tube. So that is it for fermentation. I'm going to set this aside, and now we're going to consider some other modes of respiration. So one of these is the oxidase test. For the oxidase test, we use what's called an oxidase dry slide. So it looks like this, and it has these 4 windows. The reagent in the oxidase dry slide is TMPD, and TMPD is able to detect the presence of cytochrome C oxidase. So this is an enzyme that allows certain organisms to use that respiration pathway, whereas if they are unable to use that respiration pathway, they give a different result. So we're going to use a plastic applicator stick to rub some E. coli onto the window labeled EC. Not a lot happened. So we're going to set that aside and grab a different applicator stick. And we're going to apply some Alcogens facialis to the window indicated by AF. And because Alcogens facialis contains cytochrome C oxidase, it reacts with the TMPD in the oxidase dry slide to produce a purple color. So E. coli is negative for the oxidase test, and Alcogens facialis is positive for the oxidase test. We used an oxidase dry slide that contains TMPD that's able to detect the presence of cytochrome C oxidase. We're also going to conduct a catalase test. The catalase test assesses an organism's ability to produce the enzyme catalase. This enzyme allows that microorganism to deal with the harmful effects of reactive oxygen species such as hydrogen peroxide. So we are taking some Micrococcus luteus and smearing it onto a slide. And now we are going to take some hydrogen peroxide and drop it onto the slide. And when we do that, we can see that bubbles are produced when we add the hydrogen peroxide to our smear of Micrococcus luteus. These bubbles are produced as catalase breaks down the hydrogen peroxide and produces water. So that's one byproduct of the breakdown of hydrogen peroxide by catalase is water. And the second product is that of oxygen. So oxygen is being formed, and that is causing the bubbling that you can observe that indicates a positive test for catalase, the detection of an enzyme that allows a microorganism to break down hydrogen peroxide, a reactive oxygen species that causes lots of damage to a cell when it's exposed. But if you have this enzyme catalase, which many cells do, it is able to break down that reactive oxygen species and help with the effects of that exposure.